All right. Thank you, everybody, for tuning in today. Our seminar speaker is Dr. Amanda Garcia, and Kai has called in from Alaska to introduce her, and so I'll let Kai take it away.
Thank you very much. And I seem a little bit unnecessary as a middleman, but Amanda's a great friend of mine. We went to grad school together at UCLA. And she's now a NASA astrobiology postdoctoral fellow at University of Arizona. And she studies paleogeobiology and early history of the Earth, and most recently the evolutionary history of ancient enzymes and the origin of different types of enzymes on Earth, and it's all extremely fascinating. She's an award-winning author and published in geobiology and PNAS, among many others, and that all speaks for itself, and I'll let you take it away from there, Amanda.
Sure, thank you. That's a very nice introduction. Thank you so much, Kai, especially calling in from so far away. So I really appreciate it. All right, let's see if I can do this right. Okay. All right, can everyone see this okay? I'll assume you can, so just
looks good.
Pipe up if you can't. All right, cool. All right, so it's really nice to talk to you all today. Really happy to be invited to speak in this seminar series. It sounds like it's a great series, and I'm excited to talk to you all about my work exploring the early evolution of nitrogen fixation. So I've been searching for different tools over the last several years, looking at different ways that I can and others can combine tools of paleobiology, which is really my wheelhouse. Paleobiology and geology is the world that I come from. Paleobiology, molecular biology, synthetic biology, microbiology, and biogeochemistry to tackle a fundamental astrobiology problem, and for me focused on the origin and early evolution of nitrogen fixation. So this is a foundational process for life on Earth, and in our search for life on other planets, it's critical to consider the potential on other worlds for the development of similar metabolic processes. So I'll be talking a bit about some of the broader concepts behind this work, my motivations, and mainly I'll be presenting actually a computational study where we investigated some of the key questions surrounding the early evolution of nitrogen fixation, in particular the coevolution of enzymes that catalyze that process with the ancient Earth environment. And then I'll close by telling you guys about some really exciting new work I and others have planned as part of our recently funded ICAR. That's an interdisciplinary consortia for astrobiology research, and our team is called MUSE, or the metal utilization and selection across eons. So more on that later. Right. Okay. So I'm really excited to talk to you all about this because there's several key questions about the history of life on Earth and in the universe that have been knocking around in my head for a good while. And many of these questions surround this idea of contingency in planet life coevolution. And so a lot of these questions, I'm sure, are near and dear to the hearts of many of you in the audience. But for example, first, given an alternate planetary environment, what features of Earth biology would still evolve? Are the most important biological processes that sustain Earth that sustain life on Earth today? Would they necessarily develop, or would they need to develop on other? And would other planetary environments even permit the origin of these metabolic processes that sustain the Earth biosphere? And so these are sort of frustrating questions because they're incredibly difficult to test directly. So we only have one example of life, so we have to be pretty creative in setting up different experiments where, potentially we might be able to, say, tweak biology or an environmental setting to see how Earth life's evolutionary trajectory might have been, swayed in different directions depending on a different set of coevolutionary events. And they're essential to our search for life beyond Earth, since we necessarily need to down-select to studying worlds. We have too many targets, which might have the greatest potential for harboring or having harbored extraterrestrial life. And these are also, I would say, necessarily historical questions, fundamentally because the only example we have of life operating in a completely different planetary environment is in the past. So we can use the ancient Earth as a laboratory to broaden our understanding of how life might function and adapt to, any given alternate environment. But how do we reliably reconstruct past biological and environmental states? So we have two broad categories of evidence here. One is the molecular record, so that's written in extant molecular diversity of organisms existing today. We can, map out patterns in this molecular record using phylogenetics. We can, to some degree, also constrain these evolutionary models in time, though that often typically requires calibrations from the other side of this illustration here, and that's on the right. We have the geologic record. So this sort of evidence, of course, includes fossils, ancient geochemistry, isotopic signatures, and proxies for reconstructing the type of environment that life would have existed in the past. But the main issue is that both of these records are imperfect in their own unique ways. For example, not all genetic evidence has survived to the present, since obviously organisms have gone extinct, so that genetic record is lost. Genes are continually edited through evolution. So signatures of certain biological properties or processes might be, lost or misinterpreted. Similarly, the geologic record can be overwritten through different geochemical alteration. Material can be destroyed, and the oldest pristine rocks and fossils are often the hardest to find and interpret. So I've spent a lot of time trying to think of ways that these two records might be integrated in such a way that the specific imperfections of each might be overcome. And it would also provide a way to independently test hypotheses that are generated from one record or the other. And it would be great if those could be tested empirically. So in my search, I have found that a great target of study are enzymes, as Kai mentioned in my introduction. The reason for this is that enzymes are that bridge between biology and geology. Many are the mediators of Earth's key biogeochemical cycles, so they cycle essential elements such as carbon, phosphorus, nitrogen, and sulfur. Many of these enzymes also tend to be very early evolved and evolutionary conserved as well. So that means that they have operated similarly enough in time that we can make inferences about how they might have impacted the early Earth environment. But because they've been around for such a long time as well, there are open questions on, well, exactly how functionally conserved might they have been in the past, particularly given a different Earth environment. And of course, because many of these important elements are limiting to biological productivity, the origin and evolution of these enzymes have fundamentally constrained the diversification and expansion of life on Earth. This link can also be illustrated even more directly. So enzymes operating within their host organisms are the direct producers of geochemical signatures preserved in the geologic record. So, for example, I have this cartoon illustrating isotopic discrimination. So with isotopes, enzymes discriminate among the isotopic pool. So I have nitrogen isotopes as an example, because we're all about nitrogen here. They tend to incorporate compounds containing the lighter isotope relative to the heavier isotope. And then that isotopic ratio is then carried down through that metabolic pathway and incorporated into biomass. So once the organism dies, preserved in the geologic record, those isotopic biosignatures can then be analyzed to be interpreted in the context of the functionality of this ancient enzyme. So enzymes can really be seen as a keystone in ancient Earth life coevolution. They're a practical target, and they're a really useful and informative target. And what's amazing to think about also is how evolutionary changes to biology at such a small scale, a molecular scale, can set off this domino effect that impacts planetary scale processes and has fundamentally transformed our planet, and potentially, if these same processes are operating on other planets, could actually be observable at a planetary scale. All right, so my interest with this kind of thought process was captured particularly with the nitrogen cycle. So pictured here, this is a cartoon of different transformations of nitrogen among biology. There's a variety of different biologically mediated processes. The aerobic pathways are pictured here at the top. The anaerobic processes sit down here at the bottom. And I've been focused in particular on one of these processes, and that's biological nitrogen fixation, or the reduction of atmospheric dinitrogen to ammonia, which is a bioavailable compound. Something that's interesting to keep in mind, though, is that nitrogen fixation is a notoriously expensive, energetically expensive process for biology. There's only one family of enzymes that can catalyze this reaction, and those are nitrogenases, which are pictured here in the upper right. And relative to their importance, they're also not very widely distributed among different organisms. So for example, for one, it's an exclusively prokaryotic enzyme, and even among that, it's only a subset of certain bacteria and archaea. There's been some discussion over whether the last universal common ancestor was capable of nitrogen fixation, and this has spurred ongoing debate. At the very least, I'm not going to get into it, but at the very least, due to the spotty distribution of nitrogenases among various prokaryotic organisms, if it was present in Luca, it certainly would have had to have been lost multiple times through life's history. Okay, so in terms of the age of nitrogenases, there is also a fairly wide uncertainty with this. Some of the initial estimates of nitrogenase age centered actually in the early Proterozoic, so maybe between 1.5 to 2 billion years or so ago. So for anyone who thinks about the Precambrian a lot, that's like yesterday. But subsequent studies have argued that, potentially it's much older. So for example, from the world of geochemistry, Eva Sturken and Roger Buick's group found isotopic signatures indicative of nitrogenase as far back as 3.2 billion years. And those were nitrogen isotopes preserved in organic matter in ancient sediments. And then from the phylogenetic realm, studies of, so for example, Parsons and others, I think that was Rika Anderson's group, recently conducted an evolutionary rate analysis that also incorporated an analysis of horizontal gene transfer events. And that placed the origin of nitrogenases potentially as far back as 3.5 billion years. So back to around, the earliest evidence of earliest morphological evidence of life that we have in the geologic record. So just as it continually seems that life evolved relatively early in terms of Earth's history, it so also seems that nitrogenases evolved relatively rapidly after the origin of life. So since nitrogenases have been around for so long and the Earth's environment has changed substantially over that amount of time, one of my guiding questions is, how have nitrogenases co-evolved with the ancient Earth environment? So taking a closer look at the enzyme itself, nitrogenase is also what's known as a metalloenzyme, which for me makes it really fascinating to study because it literally is the encapsulation of biology meets geology, which is what I've been searching for so long. And so this means that it incorporates metals in order to catalyze the reduction of nitrogen. So nitrogenase happens to bind one of the most complex metal clusters in biology, which is also pretty exciting. In most nitrogenases, this is called an iron molybdenum cofactor, and it's pictured here. It's essentially an iron sulfur cluster. It has a central atom of carbon, which was under debate for quite a while in nitrogenase circles, and it also incorporates an atom of molybdenum pictured here in green or teal. And the whole cluster is also accompanied by this organic homocitrate ligand. So nitrogenase, basically, the whole complex pictured here on the left, it functions as a conduit. It conducts electrons from the end here down to this active site where this cluster sits, and it's at that cluster where the reduction of atmospheric nitrogen to ammonia occurs. So the cluster itself is integral to that process and its structure. So like I said, these molybdenum-dependent nitrogenases are by far the most common and most catalytically efficient. Nitrogenases have this kind of annoying side reaction of generating hydrogen And so that ratio of nitrogen to hydrogen is typically seen as a measure of its efficiency. But it's just one form of nitrogenase, even if it is the most common. There are other forms of nitrogenase. So one, for example, is the vanadium nitrogenase. Structure of the cofactor is very similar, except for the replacement of an atom of vanadium in lieu of molybdenum. And the third form incorporates only iron. So it's just an iron sulfur cluster. So an iron atom sits here in place of vanadium or molybdenum. All of these different nitrogenase forms are operated by unique genetic pathways, and they all happen to actually be evolutionarily related, which then leads to, at least for me, a natural question: what was the metal dependence of the earliest nitrogenase? And so this is a really cool question that ties all of these ideas about planetary coevolution, because the environmental abundances of these various metals that nitrogenase uses have changed as a function of the progressive oxygenation of the Earth atmosphere. So in oxygenated waters today, molybdenum species are more soluble. And it turns out actually that molybdenum is, I think, one of the most common trace elements in the marine system. And that solubility decreases in anaerobic waters.
This weekend.
So if you go back prior to the Great Oxidation Event, prior to about 2.5 billion years, estimates of molybdenum concentration from studies of geochemical studies of sediments suggest that molybdenum concentrations may have been so low that it's inferred that they may not actually have been capable of sustaining molybdenum-based nitrogen fixation. So then that leads to the hypothesis: perhaps the earliest nitrogenases, which may very well have evolved prior to the GOE based on current estimates, may not have bound a molybdenum-containing cofactor. For example, in another scenario is that ancient nitrogenases could have been that only iron form, because iron, of course, as opposite to the chemistry of molybdenum, would have been much more abundant in Earth's early oceans. My approach to tackling this question was to use a strategy called molecular paleobiology. So this is the reconstruction of ancient proteins in order to study the evolutionary history of ancient nitrogenases. So I'm presenting work that we published last year in geobiology, and what the study was about was I reconstructed ancestral nitrogenase enzymes in silico in order to infer their metal dependence. And this is then basically a stepping point towards our experimental stages of this overall project, which I'll get into later. All right, so first, what is molecular paleobiology? The most fun and exciting way of explaining this is by calling it molecular time travel. So put simply, I can take modern protein sequences, apply models describing the evolutionary process, in order to, quote unquote, travel back in time and infer the sequences of their protein ancestors. So a lot of this probably sounds very similar to traditional phylogenetics, and phylogenetics is a big portion of this, but it's different. The overall strategy is different in key aspects. So, and specifically, if you're trying to understand the phenotype of an ancestral enzyme. So typically, in order to infer phenotypic ancestral traits based on phylogenetic approaches alone, a lot of it relies on parsimony, or kind of the simplest model. So you look at the branch patterning of a tree, traits of modern proteins, how they're distributed among the tips of that evolutionary model, and infer the ancestral trait that produces the simplest model of evolution. The drawback here is that this doesn't incorporate any specific knowledge of how molecular sequences change over time. And we have that knowledge, so it's definitely something that we can incorporate. And also, as with all models, the simplest one is not necessarily the most accurate. Evolution can do funny things. And this is particularly challenging as well when you're dealing with quantitative traits, or traits that exist continuously. So, for example, ones that aren't simply a binary of has trait or does not have trait. And finally, through molecular paleobiology, you can do what phylogenetics cannot, which is use knowledge of how molecular sequence connects to structure and function of an enzyme to then inform those inferences regarding an enzyme. And importantly, again, I'll get to this later, that can be taken to the experimental realm and empirically tested. That's the real benefit of this approach. So I got actually involved with this type of work pretty early during my PhD. So I conducted a project where I investigated the temperature stabilities of reconstructed ancestral enzymes. This was motivated by a long-running debate in the geological community on the temperature of the early Earth surface environment. So there was quite a bit of isotopic geochemical evidence. So for example, Paul Nauth and Don Lowe's work suggesting very strikingly hot early ocean, and that's represented in this figure by this gray trend line in the back. So I wanted to test this hypothesis using a completely different set of evidence, genomic evidence, and I reconstructed enzymes in the laboratory and tested their temperature stabilities, and in particular enzymes that would have belonged to ancient phototrophs or photosynthetic organisms. And the rationale being that this would then capture an ambient Earth surface environment, right? Because they would have to access light, as opposed to, say, a deep sea, hydrothermal vent or something like that. So we conducted the study. It turned out that protein stability actually increased with age of the protein, which we then interpreted to reflect an ancestral adaptation to a hot early Earth environment. This work ended up getting published in PNAS. There's intricacies to the story. There's more to discuss here. If you're interested, I can talk about that more after the talk, or you can send me an email, but I'll leave it at that for now. So it was these sorts of studies, not just my own. There's been a variety of other studies that have applied this molecular paleobiology approach to astrobiologically relevant questions such as temperature stability. It's through this work where I was inspired, I guess, to apply this to studies of metabolic evolution. So molecular paleobiology put into practice, it does follow at first like a phylogenetic study. So you collect a data set of modern protein sequences, like I have illustrated here on the left. You align them to infer patterns of homology or evolutionary relatedness between them. You then build a phylogenetic tree, which is at its root an evolutionary model. But again, the next step is that I can target particular nodes of interest. These nodes are representative of points in time or ancestral molecules that then gave rise to these descendants. I can target particular nodes of interest within that tree that are significant in terms of nitrogenase evolution based on the overall evolutionary pattern, and then directly infer the ancestral sequence corresponding to that node to some degree of statistical uncertainty. And you can also test this across different models, different phylogenetic trees, in order to kind of assess the amount of uncertainty that you have with this inference process. So I reconstructed a comprehensive evolutionary history of nitrogenase proteins. So this tree that you see here on the left, this was built from the concatenated or stitched together protein sequences of nitrogenase subunits. In the case of nitrogenase, there happen to be 3 unique subunits that are each encoded by different genes, H, D, and K, that are present among the genetic systems of the different nitrogenase forms. And what jumps out first is that in blue, right here on the left, all of these are molybdenum nitrogenases. So like I said, these are the most abundant and well represented. They themselves are actually segregated into 2 main groups. So at the top, you have a clade primarily dominated by anaerobic bacteria and archaea. And a separate clade at the bottom, these are primarily made of aerobic and facultative bacteria. But those are the common molybdenum nitrogenases. What the interesting part of the tree is up here at the top, which I've blown up for you in the center of the screen. It's actually here we have, I think, the more interesting story. So it's within this lineage where the alternate iron and vanadium nitrogenases branch. And so those are pictured here in red, iron and vanadium below it. Basal within this lineage are certain uncharacterized nitrogenase homologs, and they are notable in that they have uncertain or poorly characterized metal dependence. So we're not really sure if what sort of metal cofactor that they bind. So therefore, I was interested in targeting ancestors that were spread across this evolutionary lineage, because I would predict that there would be some sort of detectable change in metal dependence that would then be reflected in the composition of the ancestral sequence once it's inferred. So with these ancestral sequences, I was specifically interested in focusing on sequence features within the nitrogenase active site. So pictured here are amino acids. It's aligned amino acids, but specifically taken from sites close to where the metal cofactor binds, and that cartoon is here on the top. In particular, there are a couple of sites, for example, this cysteine amino acid, this histidine residue, that actually directly coordinate that cofactor and keep it in place. So not unexpectedly, there are some amino acids within this zone that are universally conserved among all modern proteins and ancestral proteins. So these are likely absolutely integral to nitrogenase function. And so evolution is pretty much telling us, don't mess with these. And that's true especially for these two residues that I highlighted before. But there are certain sequence features that I found correlated well with the different modern protein forms, so the molybdenum, the vanadium, and the iron. At first observation, I compared the sequence identity of ancestral enzymes within the active site to different modern proteins. So on the left here, this is just a matrix of sequence identity. So lighter colors here indicate lower similarity, and darker colors indicate higher similarity. So what I found was that the oldest ancestors, so for example C through E, actually resembled modern nitrogenases more so than the iron or vanadium modern variants. To extend these observations, we built a machine learning model. It was a support vector classifier that we trained on the active site sequence composition of modern nitrogenases. So this approach confirmed that nitrogenases could be reliably classified on the basis of their active site sequence composition, and it also confirmed that these oldest ancestors did actually classify as molybdenum nitrogenases as opposed to the alternate forms. Importantly, this approach also incorporated the statistical probability of the ancestral sequences themselves, and we conducted tests on this that spanned multiple alternate phylogenetic trees as well. So we have this observation: sequence features of ancestral nitrogenases, then it seems suggest a molybdenum-dependent ancestry. So this is really at odds with what you might expect if you assume that environmental geochemistry plays an essential role in determining biological metal selection. But I wanted to pull one more thread, which was to investigate the role of cofactor biosynthesis genes. So nitrogenase cofactor biosynthesis actually requires a larger network of proteins to get properly assembled and function correctly. And one aspect of this biosynthetic pathway is the formation of a metal cluster. So a simplified version of this is that the cluster precursor is formed by this protein. It's called NIFB. The cofactor, this precursor here, which is essentially that iron sulfur core of the complex, that precursor is then loaded onto a protein complex made of happen to be called NIF-E and NIF-N proteins. And it's here where that molybdenum atom is incorporated into the overall cluster. So at that point, the cluster is matured and then gets transferred to nitrogenase to complete this process. But what was interesting was that in the lineage that leads to these alternative iron and vanadium nitrogenases, several organisms represented in the tree were actually lacking one or more of these assembly proteins, so missing either that NIF-E or NIF-N protein. So if you just look parsimoniously, you might then infer that the oldest, and because these organisms tended to branch most basal within this lineage, you could then parsimoniously infer that these oldest ancestors may have similarly lacked a complete set of these assembly proteins. What's interesting though is it seems like these organisms don't mind at all. They are capable of fixing nitrogen without that complete pathway. And some of these taxa also tend to be thermophilic, which is sort of notable. So just like with these uncharacterized groups, the older ancestors may have also been, perfectly functional without the full biosynthetic pathway. So we have these two inferences. Sequence features of ancestors suggest molybdenum dependence, but they may have lacked that canonical pathway to actually synthesize the molybdenum cofactor. So I and my cofactors, we propose that perhaps these earliest nitrogenases, they were in fact molybdenum dependent, but like those basally branching, modern, uncharacterized counterparts, operated by an alternative or simplified pathway for molybdenum incorporation. So we hypothesize that perhaps earliest nitrogenases, like these uncharacterized counterparts, may have initially operated with a much simplified pathway that potentially was less specific or less efficient with that incorporation of molybdenum. And through the completion of that biosynthetic pathway, you get the refinement of that whole process of incorporating molybdenum. And this then leads to the much later diversification of iron and vanadium forms of the enzyme. So I'll take a moment here to also let you guys know that I'm working. It turns out these assembly proteins, these NIF-E and NIF-N assembly proteins, they are also evolutionarily related to nitrogenase. So they're close homologs of the nitrogenase enzyme. And so I'm working right now to conduct an evolutionary study investigating the evolutionary relationship of these two different complexes, which you can expect the preprint for that soon. So keep an eye out for that. All right, so I know I called this talk Laboratory Resurrection. Everything I've talked about until now has been in silico. So I'm going to talk now about the next steps to actually test these hypotheses in the lab. So I previously showed you this general molecular paleobiology framework here on the left. These are all the computational steps, but it doesn't have to stop there. So as with my PhD work, reconstructing ancient enzymes in the lab to test thermostability, we can experimentally synthesize the genes that encode these proteins and express and characterize them on two different levels. So we published this strategy in Free Radical Biology and Medicine a couple years back to delineate the process of resurrecting ancient biogeochemistry in the lab. So with nitrogenase, we can look at the enzymes themselves in pure form, understand their biochemical properties, for example, those correlated with metal dependence, including substrate specificity, catalytic efficiency, and on a separate level, we can assess the function of nitrogenases within a living cell. So we're currently working to engineer these ancestral nitrogenase genes into modern bacteria, which natively express their own nitrogenases, so basically replacing them. Then the impact of these ancestral nitrogenases, the function, can be assessed at the physiological level, for example, in terms of organism fitness, and important for biogeochemistry in the production of chemical biosignatures that are left in the geologic record. So for example, signatures of nitrogen isotopes. And so basically that closes the loop that I presented way back at the beginning of this talk, uniting and independently testing hypotheses that are associated with the early evolution of nitrogen with systems deriving both from the genomic and geological records of life. So for the nitrogenase system in particular, I'm using a model bacterium called the Zotobacter finlandii. It's a nitrogen-fixing aerobic proteobacterium. It's really great. It's practical and it's useful in that it possesses genes for all three metal-dependent forms of nitrogenase at once. So this makes it then possible to set up really interesting experiments regarding the evolution of metal specificity. So we can tweak this organism in different ways to kind of set up different experiments. We're in the process of developing and refining this system. I'm going to say it's too early to share the outcomes of this with you today, but do keep an eye on our work in the future. I do, though, want to highlight some recent work that we published in Current Protocols in Microbiology. So this was work led by some really great students that I'm working with at the University of Arizona. We have Brooke Carruthers up here on the left, who is lead author in this study. She's an undergrad, and we have Alex Riviere, who's currently a master's student. So we worked to develop a high throughput automated method for assessing nitrogenase function in engineered Azotobacter, which we are currently applying to our work now. Okay, so finally, I want to talk a little bit about our other upcoming exciting work as part of the ICAR Muse team. Batul Kachar at the University of Arizona is leading this as principal investigator. I'll be serving as a co-I, specifically leading the team concerned with nitrogenase laboratory resurrection. So the nitrogenase story is central to Muse, and it's one piece of a broader question, which goes back to what I talked about on my first slide about contingency and planetary coevolution, and how that can inform our search for habitable and inhabited worlds. So, for example, what if we are confronted with exoplanetary data suggestive of a robust nitrogen cycle, maybe from some spectral observations, but also the scarcity of necessary metals for biological nitrogen cycling? So would we actually evaluate that as a suitable target for further exploration? And the reason I bring that question up is that if you go back to where I was presenting the story of nitrogenase, if I told you that most nitrogenases use molybdenum, and then you saw a snapshot of early Earth marine geochemistry, you might very well write off Earth as unsuitable for a robust nitrogen cycle. But of course, that would have been a false negative. And so there's nuance to these sorts of investigations, and it's imperative that we think about these sorts of things, since the suite of potential targets of exoplanets is expanding so rapidly, and of course, that's only going to increase exponentially. So within this team effort, my strategy, like with everything, is to reverse the clock. So this is a framework we published recently in Chem Biochem. The main objectives of this sort of work is to determine what factors during a planet's evolution determine the suite of bioessential elements. So that includes, chance, carbon, hydrogen, oxygen, nitrogen, sulfurous, phosphor, that good stuff, as well as trace elements. And does that suite of elements change through time? And to what extent can life adapt to use alternatives as the availabilities of those elements change over a planet's history? And so this mission essentially is to determine what does life want. We can explore what drives the natural selection of chemical elements. Is it biological function? Are some elements just that good at doing what they do in a biological context that life will just find a way to use them? Or is it more constrained by environmental availability? Does life necessarily have to reflect the surface chemical composition of a planetary environment, just based on what is available? And so nitrogenases are really the perfect target to investigate this, because they play a role in making available one of the most essential elements, of course, nitrogen, and they have that connection with metals to variation in abundance of trace elements that can occur over a planet's evolutionary history, as has occurred on Earth. And so all of this will be used to directly inform the next generation of life detection missions. All right, so I'm going to wrap up. With that, I'd just like to acknowledge my lab, the Kachar Research Group. If you're interested in learning more about the different projects we have going on, please visit ancientbiology.org. I'd like to acknowledge my collaborators who have been involved both with the computational and experimental work for this. Finally, my funding sources, University of Arizona, especially NASA Astrobiology postdoctoral program, NSF, and the John Templeton Foundation. So thank you very much, and I am looking forward to answering any questions.